Okay, welcome back to Anatomy and Physiology here with your professor, Dr. Sean Clovell. Today, as we continue talking about the organic molecules associated with the human body, we come to our final category of organic molecules that we'll be discussing throughout the semester, and that is the nucleic acids. This is DNA, RNA, and ATP. DNA and RNA are actually some of the largest molecules in the body. They contain carbon, oxygen, hydrogen, nitrogen, and phosphorus. Now, the monomer of a nucleic acid is the nucleotide, and each nucleotide really contains three basic components. So, the first component of a nucleotide here is the 5 carbon sugar. So you can see that there's a 5 carbon sugar there. That 5 carbon sugar is either ribose or deoxyribose. So remember that there's those pentose sugars. Those pentose sugars are either deoxyribose or ribose. So, as you can see in the example that we have here on our screen at the bottom here, that's deoxyribose sugar. That's the pentose sugar. There's also a phosphate group, so there's a polar phosphate group here that it has a charge, and a nitrogenous containing base. So a nitrogenous containing base here as well. So the building blocks of nucleic acids are the nucleotides, which is composed of a nitrogenous containing base, a pentose sugar, and a phosphate group. DNA, one of the classic nucleic acids, is formed of a sugar phosphate backbone. So the sugar, the pentose sugar, and the phosphate groups alternatingly are associated with each other to make up the outside of the ladder here. And then the nitrogenous bases can be one of 4 bases: adenine, thymine, guanine, and cytosine. Adenine always associating with thymine, guanine always associating with cytosine. And so the rungs of the ladder that are associated here are complementary. They're not identical, they're complementary. So there will always be a complementary base that associates with them. Humans have almost 22,000 genes in their DNA locked up in the 46 chromosomes inside the nucleus of each cell. These genes can carry the genetic code to actually build one's body. On the other side, RNA consists of a single strand of sugar phosphate backbone studded with these bases. And again, there's you can see that the some of these are actually the same: cytosine, guanine, and adenine. And as opposed to thymine, we have uracil. Which brings us to some of the differences between DNA and RNA. And the first is, as we can mention here, the sugar group. Remember that the sugar group contained in DNA is deoxyribose. The sugar group contained in RNA is ribose. So the first difference between the DNA and RNA is the pentose sugar associated with them. The second difference is the nitrogenous bases. So if we zoom in a little bit more here, we'll see that the DNA contains the nitrogenous base uracil. Excuse me, let me say it the other way. DNA contains the uh, nitrogenous base thymine, while RNA contains the nitrogenous base uracil. So there's different nitrogenous bases associated with it, as well as in the basic structure, right? As well as the uh, different pentose sugar. The number of nucleotides. So DNA is much larger than RNA. RNA is actually just a small portion of DNA that could be copied to help make a protein. So while DNA is the entire cookbook, the RNA might be the recipe to cook one specific dish. The shape is different. Obviously, we mentioned the size, but also the double helical structure. So RNA really just has a complementary strand to DNA. So there's a complementary strand to DNA, cytosine matching with guanine, uracil matching with adenine, and as a result, what you get is a portion of the DNA, whereas DNA has the double helical structure. Then finally, the function. We've talked about the function where DNA houses all of the genetic information to make all of the proteins in the body. RNA can take a small portion of that and then send it out to the cytosol into the cytoplasm to make a specific protein. And then our final but not last but not least, right? Uh, Nucleic acid is that of adenosine triphosphate. This is the energy currency of the cell, and the energy is contained within these high energy phosphate bonds. So, adenosine triphosphate contains the nitrogenous base adenine, the pentose sugar ribose, and then three phosphate groups held together in high energy bonds. And so when energy is released when the phosphate is hydrolyzed, meaning that the bond between those phosphate groups is broken, energy is released, and as a result, the product is adenosine diphosphate. If that another phosphate was hydrolyzed from that, we'd have adenosine monophosphate. And then the adding on of phosphates is referred to as phosphorylation. That's what happens in the mitochondria. In the mitochondria, ultimately, we rephosphorylate these ATP, and that's why it's called the energy or the powerhouse of the cell. That's where the energy of adenosine mono and diphosphate actually is rephosphorylated or phosphorylated to get that phosphorate group back. So that wraps up our section on organic molecules. And as we finish talking about nucleic acids, remember DNA, RNA, and ATP. Remember that the primary structure of the of our nucleic acids is the nucleotide. It contains a nitrogenous. Well, let's start with the 5 carbon sugar, a nitrogenous containing base, and a phosphate group. It'll be important to identify the differences between DNA and RNA, as well as the structure of ATP. Again, thanks for your time. I look forward to seeing all your responses through all the assessments and feedback. Again, have a safe day and take care.